imagine you're traveling abroad, you walk a lot, you visited a lot, and now you're hungry and it's time for lunch. So you are going to this food street market, and while the food really looks good, it's not very clean, but it will be fine. However, 2 days after, it's not so fine because you start to have cramp, fever, fatigue, and a severe bloody diarrhea that make you create a special bond with your bathroom for the next few days. So you might have just met a tiny but dangerous bacterium called Shigella. Shigella is causing more than 200 million contamination every year in the world, with more than 200,000 deaths, and it's mostly kids under 5. Why it's happening most of the time in low-income countries where you have poor sanitation is not just over there, because in the US we also observe more than 400,000 cases every year. So now why studying Shigella? So it's very easy to transmit the bacteria because only 100 is enough to get sick, which means that you can infect easily your friend and family. We don't have any preventive treatment such as vaccine, and the antibiotic treatment are not very efficient, and we observe an increase of strain presenting resistance to this antibiotic, which makes Shigella a public health concern. So what can we do? I believe that better understanding how Shigella is infecting us and how our body responds to Shigella is very critical to find new therapeutic target to treat infection by Shigella. So during infection, so after ingestion of contaminated food or drink by Shigella, the bacteria is able to resist to our gut, and what is really the last part of it, called the colon, here in red, it can infect it and multiply inside it. Now, if we do a zoom on it, and I apologize if I wake up any biology class trauma, but we know that the colon presents colonic cells that are really tightly close to each other, tightly attached close to each other, and here in blue. And we know that Shigella is able to enter into this cell, multiply inside it, but also disseminate into the adjacent cell, leading to a massive recruitment of immune cells, leading to this bloody diarrhea. So we know that the capacity to disseminate into this layer is very critical for infection, and we know that if we block it, we could eventually reduce the infection by Shigella. And that's where my research comes in. So in the lab, we are mimicking this cell layer by growing human colonic cell in a dish, and if we wait enough longer, we can generate this layer of cells that we can infect with Shigella, here in red. And over time, the bacterium can divide and disseminate into the adjacent cell, generating an area of infection called focus of infection that we can measure and compare according to different conditions. So I used that for the first step of my project, which consisted to test over 400 different chemical compounds. We know that this compound targets specific proteins that our body is producing. And one of them was really interesting for us. It's a compound that blocks the function of one protein called PIK3D3. And as you can see here on the right, when we use the chemical compound, the area generated by Shigella is smaller compared to a normal condition. The second step was to study in detail the function of this protein. And for that, we use time-lapse and fluorescence microscopy. So I was able to track individually each bacterium going from one cell to another, and in order to better study this function. But I will not go too much in detail. It will be kind of boring for you. And then the last step was to move from the lab from an actual in vivo model, where here the colon is represented in green, Shigella in red. And as you can see, when we add the compound, the area generated by Shigella are smaller in this green area. So this result is very exciting because it really shows that targeting some protein that our body produces can be a way to treat Shigella infection. So next time you are going on a culinary adventure, remember we are trying to keep your gut safe. Okay.